Can you hear me?
Yeah.
Okay, great.
Good morning, Marcos.
Good morning.
Yeah. How are you?
Good.
Yeah, good morning, Dr. Marcos.
Hi, how are you doing it?
Hello, Marcos.
Hi.
How are you?
Good. Hi, Anil, killer.
So it's time to start now, I think. Good morning,
Yes.
one and all.
Good morning, everyone.
Morning.
Welcome to today's inaugural session of the webinar series on diverse areas of biotechnology, hosted by the Department of Bio and Nanotechnology of Guru Jambeswar University of Science and Technology, Hisar. My name is Rakesh Yadav, and on behalf of the organizers, I welcome Honorable Vice Chancellor Professor Tankeshwar Kumar, keynote speaker Professor Marcos Egea-Cortines, Chairperson of the Department of Bio and Nanotechnology, Professor Vinod Chokar, Professor Ashok Chaudhary, other faculty members, dignitaries, and the participant audience. Before we go further, I would like to request Professor Vinod Chokar to give opening remark and overview of the webinar series. Welcome, sir.
Hello.
Yes, over to you, sir.
Yeah. Respected Honorable Vice Chancellor Professor Tankeshwar Kumar, keynote speaker Professor Marcos, faculty members, and friends. I welcome you all to this one-week webinar series in the diverse area of biotechnology during August 25th to 31st, 2020. More than 19 places.
I'm not sure. I'm hearing anybody. Vinod, I didn't hear your last words. So do you think, should I start?
I could not listen to,
Yeah, okay.
Sir, please unmute your mic. Professor Vinod Chokar, please unmute your mic, sir.
I could not listen to him also.
Okay. Can everybody hear me? Or most people?
Yes, Professor Marcos.
Okay,
You are
so
audible.
I'm going to start my lecture with something.
Professor Marcos.
Yes. Do you see my computer?
Yes.
Yes, yes. Before starting presentation, Professor Marcos.
Yes.
I would like to request Honorable Vice Chancellor, sir, to address the participant to give the valuable inputs.
Rakesh.
Yes, sir. Yes.
Yeah, I am continuing. Yeah.
Yes. Yes. Continue, sir,
I
please.
was not audible at that time.
Yes. Yes.
Yeah.
Please, sir, continue.
Yeah. But there was interruption in between. Yeah, I would like to welcome all participants to this webinar series in the diverse area of biotechnology from August 25th to 31st. More than 14 scientists from India, Spain, USA, Canada will deliver a lecture in the diverse area of plant biotechnology and animal biotechnology, microbial biotechnology. Our department is conducting this type of lecture series every year by inviting scientists from India, and that's true from the nearby places like Delhi, Chandigarh, Haryana. speaker from the abroad. And it is an honor for us to have Professor Marcos among us. That Professor Marcos is presently working as a director, Institute of Plant Biotechnology, Technical Institute of Cartagena, Spain. He did PhD from Israel in 1997. Then he joined Technical Institute of Cartagena, Spain in 1999. He did 2 postdoctorate, one from Purdue University, USA, and another from Max Planck Institute, Germany. Professor Marcos is not new to this campus. He has already conducted a GJAN course, and he was very popular here among the students and the faculty member of the department. I indeed fortunate to welcome Professor Marcos, and I would like to express my sincere gratitude to Professor Marcos for delivering a lecture on the regulation of scent emission by floral organ and cloak gene. It is my privilege to extend a warm welcome to Professor Tankeshwar Kumar, Vice Chancellor, Guru Jambheshwar University, Hisar, as a chief guest. He is a very soft, kind-hearted, and possessing positive attitude towards everyone. He is a source of inspiration and guidance for us, and he is a pillar of strength to bring this department at this stage. I am happy to inform you that MSc Biotechnology program of this department is supported by Department of Biotechnology, Government of India, under his able guidance. Under this scheme, DBT will provide more than 1 crore every year, and students will get fellowship of 5,000 per month. I urge all the BSc third year students of our university also, and to the other students also, that they must apply for this Gate B examination, as the last date is still August 30. Although we have organized this webinar for the department only, but by knowingly or unknowingly, this process I hope you will enjoy this academic session over the next week. And thank you, everyone.
Thank you, sir, for the opening remarks and overview of the webinar series. We now request our Honorable Vice Chancellor, sir, to address the participants to give valuable inputs. Welcome, sir.
Thank you, Professor Vinod Shokar, and Chairperson of the Department of Biotechnology. I'm really happy that you are organizing a webinar series involving speakers from Spain, Australia, and USA, and across the India, and the participants from across the country are participating in this diverse field of biotechnology. I hope that this webinar series will be very useful for not only for the younger students, but also for the experts of the field. And I'm today really thankful to our today's first speaker, Marcos from Technical University of Cartagena, Spain, and I'm really happy. I could see the first slide, with a lot of spices. I could recognize these spices from some of these things we daily enjoy, and these are the, maybe we call it as Indian spices. I don't know whether it's all, but it is available globally, we know. But these are the spices we use it. And these days we talk about a lot of things about these things, because we have to improve our immunity in the time of the COVID-19, and all these things are happening. And biotechnology, I say every field is now see that when we have the different new normals, every field is important, whether it is computer, biotechnology, or even engineering, different kind of devices we have to make, in addition to the medicine and all these things. So everyone has to work what he can do for the new normals and things like that. So I'm really thankful to our the speakers which are today and maybe tomorrow and so on, and all the participants. I wish really success of this webinar series. I congratulate Vinod Chokar and Shok Chaudhary, and also I see Vijay Yadav, Neera Dilbagi. I could see, I really congratulate all of you and thank you very much. Thank you.
Thank you, sir, for motivating us for this webinar series. We are all inspired by your great words. So without any further ado, let's move to our today's webinar. Just a reminder for audience, if you have any question during the presentation, please type them into the chat box. I'll bring them up during the presentation and we'll have a time for question at the end. So I'd like to kick off things by welcoming Professor Marcos. He is really a great speaker. I'm sure we'll enjoy his presentation. The floor is yours now, Professor Marcos. Over to you, sir.
So thank you so much. I hope everybody's hearing me. And Professor Tankeshwar Kumar was right. Today is a special day for me, and I want to bring the beginning of my seminar about something which goes into the heart of the Indian people. What you see here is a picture of something that people like you know very well, which are the different spices that are used to create what is called a garam masala. I guess I don't have to explain to you what is a garam masala, but maybe yes, I have to explain you a garam masala as a basic concept out of which I'm going to build up the seminar, which is scientific, but it has several important concepts for life, not only for the specific aspect of volatiles. So today is a special day because I married on the 25th of August many years ago. And one problem in India is that you say, oh, I cannot marry somebody from the north because the people from the north do a different garam masala. I don't like that garam masala. I like the one from Uttar Pradesh, which is different from the one that they do in the south. And then no, but the other one comes from Punjab, and that is again different. So what is garam masala as a whole? So I did a small analysis. Added in a certain place of the country. Somebody will raise their hand and say, oh no, my mother is adding something else, which is true. But this concept that you have a core garam masala and then you can add different things and that creates the diversity of the spices in your country. That is what I'm going to be talking about today from a molecular and genetic perspective. So the title of my talk is Constitutive and Non-Constitutive Scent Profiles. And how do you coordinate that to create the profile of the scent of a flower? Since this seminar is going to be followed by undergraduate students, okay, there is here a set of concepts which I'm going to be walking through the seminar so that you know what we're going to be talking about. By the way, if you need the seminar, I will send it by mail to Professor Chokar so that you can have it and you can distribute it to your students and to Guru Jamshedpur University staff if you want. Okay, so it's yours. So the basic concept I'm going to talk about is what is a housekeeping gene and what is a differentially expressed gene. So think about the core garam masala, the core genes which you find in every cell, and those which are specific for a certain type of situation. I'm going to explain what is a differential gene expression and what are the functions of differential gene expression. As an example, I will talk about asymmetric cell division and development, because as you will see, that plays a key role in everything. I'm going to explain very slightly what is the concept of organ identity. How do you organize systems and how do they cope with the environment? We can think about plants and animals as organisms which are subject to different times of the day. Temperature is going up or down. Nice way of how you should smell. Okay. So let's think about for a second about how do cells function. And let's think about bacteria, or here we have depicted a plant cell. So bacteria and plant cells, they can do different things. They can grow. Sometimes they don't do it. And for growth, you need to uptake your nutrients. You need to produce energy. You have to degrade sugars. You need to produce your cell walls in case of bacteria. You need to produce cell walls in plants. You need to create new membranes. If you are dividing, then you have to do your DNA replication, right? So if you think about it, a cell has to have certain standard functions which are working all the time, because that is what makes a cell to be alive. Okay? So if you think deeply, probably when you look at a cell, from different types of a body, or from a plant which is a cell based. That will be part of the genes. But most cases, housekeeping genes are those related basically to metabolism. Think for a second about the central nervous system. Their cells are not growing, they are maintained. Think about a tree. The cells which are and finally, you have several enzymes. I wrote here just a synthase, a permease, a transporter, so that it's easier to follow. And these enzymes are expressed, let's say, in every cell, right? So we can say, oh, these are housekeeping genes. And this is uptaking glucose, is letting the glucose to be passed into the mitochondria, and then it's producing energy, and then the cell is alive. But not all genes are housekeeping. Genes are sometimes expressed differentially, and these are extremely important. Now, I'm give you in the next slide a simple, nice example. What you see here on the left is something which always makes me smile, and I think it was also degradation, they are not expressed all the time. They are only expressed when lactose is present. Here is a nice example. It's a typical example from a book. This was worked out by Jacob and Monod, and they obtained the Nobel Prize because this is how they explained gene regulation. So what you see here is that you have a repressor which is binding to DNA. And when lactose comes in, that repressor comes out of the DNA, and the genes involved in lactose degradation are activated. So this would be a very nice example of genes which are not housekeeping genes, are bacterial genes which are expressed at a certain time, at a certain point, in this case, in response to an environmental condition, which is that we had this thing. By the way, I hope my seminar is not making everybody very hungry, or maybe yes. So, cells which have differentially expressed genes. We see here on this diagram the expression of genes, which is here depicted by this arrow. And here you have the amount of RNA which is produced from this gene and then from this one. As you can see from this one, nobody's producing anything. And you have transcription factors that recognize these elements on the DNA and they're going to activate these genes. And then you have this cell next to it, which is different because the transcription factors it has are different. So you see that this cell is producing these gene products. As you can see, there is a difference because this one is not found in this cell and this one is found in this other cell. And this is all revolving around these transcription factors which are binding the promoter of these genes and activating these genes, right? So typically, okay, and this is a wrap up, housekeeping this concept was built up upon the experiments of the scientific community. So if you come and ask yourself, what are the differentially expressed genes, let's say, in a flower? What are the differentially expressed genes between a flower and a leaf? Well, something obvious is that leaves are green. So we're going to have chlorophyll genes in the leaves, which are not expressed in the flower. But then there are other ones which are going to express in every cell. And again, this is a matter of doing experiments and finding who is housekeeping and who is not. Right. So as I was mentioning, okay, differential gene expression is extremely important for everything. Let's think about an example here, which is about developmental biology. The developmental genetics of an organism, which is the field that I do most of my work, asks a very simple biological question. What are the rules and the laws that create an organism with a phenotype, right? And how does it happen? So if you think about a zygote produced by a sperm cell and the oocyte, or by a So when it divides, the cell which is attached here has a set of RNAs. And the second cell, which is the daughter cell, is different through the mother cell. So this is what is called an asymmetric cell division. Here you see that the two cells become different. So this one is going to be becoming a different cell type. It may give rise to a different organ, right? And that happens by physical signaling between these two. Here you have again another situation which is slightly more complex, and that is that you have here a set of gradients where you have things happening on the right and things happening on the left. And the cells on the right are giving a signal, so this cell dividing here ends up being different from the mother. Of course, all these cells have a set of genes which are common, but this one has a different set of genes which is going to be differentially expressed and is letting this cell become different. And last but not least, we have the same concept when there is no signaling around. That happens just because that cell has an intrinsic asymmetry. And this asymmetry doesn't happen from nothing. You can ask me, typical student question, Marcos, why is it asymmetric? And the answer is that the messenger RNAs which are deposited on this side of the cell are different from the ones deposited on this side of the cell. And when they divide, the set of messenger RNAs which you get are different. And that is letting this cell become something new. Okay? So you have an intrinsic separation of messengers. So the take-home lesson is that differential gene expression explains and underlies most biological processes you think about, right? So I'm addressing here the basic students. When you look at biology, you should always think, okay, what are the common aspects here? Remember the garam masala example. What is common to everybody? And what is different? Okay? So use that critical thinking to address and identify the difference from the common. And then you will know what makes that a specific process, who is controlling it, and how is it coordinated. I'm going to have petals of different colors. Then you're going to have inside a number of stamens, and then in the middle you're going to have the carpel, which is the or gynoecium, okay, which is where you have the female floral sex organs. Now, how does it happen that all plants have the same structure of the flower? That happens because you have combination of genes which are giving the identity to each of the organs. So we have protein complexes, which are the so-called your stamens, okay, different one for the carpel and different one for the gynoecium, but they are all combinations of proteins which are together binding DNA and forming this tetramer, and is giving you this structure, right? So what is the take-home lesson here? These are differentially expressed genes for each organ, and they actually create a cascade of events giving you a nice, beautiful flower formed, okay? And this beautiful flower is formed by a combination of genes which are activating downstream genes. So one important aspect here that I want to tell you, hierarchy from a personal perspective. Genetic models are not based on the ideas of scientists coming from nowhere. They come from the analysis of mutants in this case. So the explanation of the phenotypes is the one right. So interesting is that, as I was saying, this model is true for everything. Every plant. You have here a rice flower. The rice flower, okay, has a palea, has a lemma, has the stamens, and has the carpels, and the organization is practically identical to something like a Arabidopsis or another type of flower. And the genes which are coordinating these different organs are basically the same. So I'm telling you, this is a general rule of set of genes which are giving you the formation of this flower. Okay. Now I'll show you a mutant which is called compacta, which we're working with. I will show you data later on, where this organ is not well formed. You see here a nice wild type, right? A wild type plant has these big petals which are very nicely formed, and the stamens are here, they are concealed. You don't want your stamens out because they dry. You see here that they dried. So if you dry your stamens, you're going to have problems to get to the next generation. So this mutant is, although nice, it is creating a big problem to the plant. And as you can see, the petals are not well developed and the stamens are out. So this is a weak B function mutant. This petal is not okay. I'm going to show you here two others. You have here another compacta allele, which is in this case in a white colored background. I showed you this because some students would say, oh, look, they're different color. Yes, I hope you these a plant. If you take a turmeric, I don't know how you call turmeric in Hindi or in Tamil, but the idea is that this is a very important plant for you. And you don't use the leaves to obtain the powder. You have to use the tubers because that is the identity of the organ driving the expression of these genes. So with this slide, you see perfectly well that when you have the proper organ identity, then you're going to produce not only the structure, it's going to be translated at the end into the specific components of that organ. Okay, so think about, let's say, emojis. If you want to have a boyfriend, maybe you might use the emoji of the rose. Okay, if you have to send it to your mother-in-law, maybe you should send the rice because that shows that you're a person that you're getting a lot of money. These are the same as I showed you. We see them as different, but from a genetical perspective, they are identical. So that's great. I'm talking about something that is happening by default, right? But if you remember the bacterial example, I gave you the bacterial example and I said that that is an example that every student should know pretty well. You should remember it. It is the basis of how gene regulation was uncovered. Okay, one of the key experiments. Another one that I'm not talking about today is about transposable elements, but it is more complex, right? But that example is based on environmental conditions. You don't take paneer tikka every day, right? Although you might eat your curd. And when you eat curd, you have the same situation. Your bacteria get lactose in your gut. So that is an environmental condition. But if you ate only paranta, then you would not. So the environmental conditions play a key role in how an organism reacts and what it's doing. I think everybody agrees. And then the question is, how do you integrate the signals from the environment into yourself? Does it happen by magic? And the answer is plainly no, it doesn't. It happens through molecular mechanisms. So the environmental conditions play a key role in the metabolic structure in plants. And think about the scent of a flower. The scent of a flower is like the personality of the flower. The scent of a plant is the personality of the plant. For you all biotechnology students, also for PhDs and even postdocs, there is a huge field of work related to plant resistant to pathogens. It is a horribly important field because many people, their life depends on having a good crop. Okay, otherwise they will have problems with hunger. I don't think I have to explain that. Everybody understands. And in many cases, weather plays a key role because plants can defend themselves, sometimes they cannot, based on the weather. And that is related to the setup of genes which are activated and repressed. Now, how do you get those genes to be activated and repressed by the weather? This is what I'm going to show you in a second. So on the next slide, you see the one that I showed you before, and that is that plants have something that we also have, which is that we react different to day and night. We humans are daily animals, okay? Other animals, let's say cats, they are night animals or bats, right? But we are active during the day, and plants have active parts during the day. Remember, during the day, you are doing the so-called light-dependent photosynthesis. And during the night, you're doing night-dependent photosynthesis, right? You're doing other processes. So clearly, there has to be somehow a way of those signals to get into the system and get coordinated so that things will work well. So I'm going to give you a molecular example. So this is getting slightly more complicated, but I want to show with this slide that, as I was saying, that doesn't happen just randomly. There are specific molecular mechanisms that explain everything. Here we have a protein called CCA1, and CCA1 is part of the so-called plant circadian clock. And CCA1 has the following situation. It has a partner, which is a protein kinase, which is, it's called casein kinase 2. There is no casein in plant, it's just called like that because it looks similar to the animal casein kinases, right? And you have different combinations of these proteins in plants. And depending on the combination, they do something different. So when temperature comes in and drops, what happens? Think about it for a second. We have a temperature in our body between 35.8 and 37, depending on the time of the day. That's why, and excuse me for what I'm going to say now, but I'm a scientist, okay? If they measure the temperature on your head, depending on what you were doing before, and depending on the time of the day, it's going to fluctuate. So we keep a very tight range of temperatures because our temperature is controlled. So if they take your temperature saying, oh, you may be COVID positive, well, it's not true. I was running. So because I was doing sport, I have a higher temperature. But think about plants. Plants might have encountered a difference of 10 or 15 degrees of temperature between the day and the night on any day. Think about Hisar. You may have today, well, I don't have my phone here, but you have probably 33, 34 degrees, right? And when it goes at night down to 24, 25. Now you go to winter and you may have 20 or 23 and 15. So how does a plant to maintain a day activity which is not changing so much from one day to the other? And they do it because they have what is called a temperature compensation. How do you compensate? Let's take this gene. When temperature drops, then the protein is going to be binding DNA faster. When you phosphorylate the protein, then the activity of DNA binding is changing. So when you phosphorylate this protein, for instance, is activating the chlorophyll binding protein. When it's not phosphorylated, and this is temperature dependent, then it's going to bind other genes, and it's going to create a different speed of the clock so that we can coordinate ourselves and slow down when temperature is high and increase the speed when the temperature is low. Because when temperature is low, all molecular functions go down by chemical laws in plants. When temperatures go up, everything goes up. But you want to maintain your day clock. So you cannot go crazy when it's hot or go to sleep, okay? And become sluggish when you are under cold temperature. So this is a way of coordinating your signaling inside the cell. Now, CCA1 is called LHY in other systems. And I'll show you here the very complex plant circadian clock. The plant circadian clock is very complex, but for you students which are used to play video games, think about it in the following way. You have what is called the morning genes, the morning window. And during the morning, these genes are activated, okay? And then they repress the midday genes, okay? When these genes get down, then the midday genes go up. This is like when you're playing a video game and you pass to a new level. So this is the morning level, and then all of a sudden you go into the midday level. And midday the so because if you don't have your clock, those scent products are not going to use very well. And I'm going to show you in the last part of my talk, how do you coordinate those genes? And I'm going to tell you something important for all you students, and especially for graduate students. You have to do the experiments. Do the experiments. Only when you do them, you know what you're getting. We found that we were always wrong. When we did the experiments, we found that what we were thinking was not correct. I ask you, before I show you the data, So what do you think happens for a flower? We think that flowers have a very strong smell, right? I guess everybody agrees with me. Flowers have a very nice, strong smell. So our intuition would tell us, oh, that means that in the plant, what you do is that you're going to produce a lot of scent the gratitude. Mass spectra of the garam masala volatiles, you would go crazy. It has more than 100 compounds. And then you go to a different garam masala and it has again 100 compounds. But as I showed you, not all of them are common. So defining what is a constitutive metabolome, like in constitutive genes, and what are differentially expressed metabolites is not a simple task. It has taken years for the community to identify those genes. And it's going to take us some time to identify those metabolites, the ones which are produced by most cells or every cell, and the ones which are produced by only by certain types of cells, okay? So this package allows you to do that, to analyze the data coming from GCMS in a very easy way. Analyze 40 samples, 50 samples, 35, 60, 100, and the system will help you. Okay, so you become more robust in your decision of what is constitutive and what is not. So the data set I'm showing you here is that when we look at the wild type and say, what are the volatiles which are found in every flower? We have only 4, which are very few. Think, oh, only 4 volatiles are found in every flower. The answer is yes, only 4. That was a surprise because we had always found a lot and we always thought, oh, everybody, they are produced by every plant. No, this is a data set of 36 flowers. If you look at the scientific literature, you'll find that only people use 3 to 5. Here we have 36 samples. And on 36 samples, you have only ocymen, dimethoxytoluene, acetophenone, and methylbenzoate. And this comes where, as I was saying, we were wrong. When you don't have a proper identity, then in this case, compacta becomes completely crazy. So one flower produces one set of volatiles, another one a different set of volatiles, another one makes yet a different one, and it doesn't have a single compound which is produced by every. One make part, which is the constitutive paranta. That was a real shock for us, that compacta does not produce a single compound. This is going to get more interesting later. And if you look at a strong loss of function, then you see that the way this flower smells is totally different. They do produce volatiles, but they are not the ones from the flower. These volatiles actually are the ones produced by, surprise, surprise, by the leaves. So it turns out that when a flower is not properly formed, it remembers that it was a leaf, and it produces a leaf-like smell. What happens when you downregulate your clock? Well, things get here somehow changed. You still smell like a flower. You are missing somebody. But I'm going to show you on the next slide something which is, okay, these are the ones which are expressed in every flower. So these are the constitutive. Now what happens to the plant, to the ones which are not constitutive? And here comes the other surprise. So here I'm showing you all the compounds produced. And we find that the wild type doesn't produce many compounds. These are produced compounds which are produced sometimes, not all the time. So one flower might produce indole and another one not. But you don't produce a lot of them. But compacta, look at that, it went crazy. It produces a horrible amount of volatiles, okay? It produces everything. The efficiency looks different because it's a loss of function, which is looking like a leaf. So you can say, oh, these are the leaf volatiles, but look at compacta. It's producing a horrible number of things. And when you look at the loss of function in the clock, you also get the same picture. Appears that the clock is repressing all these compounds that do not appear in the wild type. Look, they don't appear in the wild type, they are here. They shouldn't be, okay? So the clock is actually repressing too many things to happen. And compacta is repressing for all these volatiles to appear. I'm going to give you another example so that you remember very well. Let's think for a second that you do your dal makhne. And instead you go crazy and you put all the dals that you have in a restaurant, you put them all together in a pot. Do you think that makes sense? And would you want to eat that every day? A single dal which has everything inside? Oh no, you don't want that, do you? You want to have the different tastes. They are different, and that's what you want. So a flower has to have a certain set of volatiles. That is what makes it a flower. You don't want all this list of compounds coming out, okay? This is a clear deregulation where this is anarchy, okay? You don't want anarchy. You want an ordered set of volatiles produced. So to end up the slide, look at this. The total amount of volatiles that we found is 58. Wild type produces a total of 10. Compacta produces 43. The efficiency is 19. And the RNA of a clock gene, this is the downregulation by RNAi, produces 30 volatiles. So this means that the coordination of the constitutive metabolome and non-constitutive is produced by the organ identity and by the clock. Because when you downregulate identity genes or clock genes, all of a sudden you produce many more volatiles. And there is something very, very, very interesting about this slide, and that is the following. If you would analyze the wild type, you would say, oh, my genetic capacity to produce scent is down to 10 compounds. However, just mutate a gene and I produce 43. So I can produce this thing called isolongifolin in antirhinum. I just didn't know I could produce it. I never find it in the wild type. I don't produce it in the wild type, or pinene. This one is easy to remember because it's a typical pine tree smell, right? But we don't produce pinene in wild types. So when you mutate genes, sometimes you uncover a genetic capacity to produce volatiles that you don't find in a wild type plant. So when you look at this list of compounds, this is very difficult to understand. I mean, think about it. This is a list which might be good for computers, but we are humans. We are not used to think like that, okay? So we want to see this in a more organized way. So here you see two different pathways I'm going to show you. And here you see when the constitutive would be on the top and the non-constitutive on the lower part. And here is what you find. So methyl cinnamate. Does anybody there in the audience, can anybody tell me or think about for a second, what does methyl cinnamate smell like? Just by the name, you may have a clue. It's a major component of cinnamon, right? So we find methyl cinnamate in all the genotypes, but it's not constitutive. Let's look at this one on the other side. Methyl benzoate. Methyl benzoate is constitutive in the wild type flower, and it's also constitutive in the clock, but you need the identity genes to produce it, okay? And interestingly, you have here this pathway, benzaldehyde, benzyl alcohol, and benzyl acetate. And if you realize, you only find this one in the RNA for clock gene, then you find the other one, benzyl alcohol, in these three genotypes, and you find the benzyl acetate again in the clock line. So this means that this pathway is repressed by the circadian clock. Because when you downregulate the clock, this pathway, the complete pathway gets activated. So you get three compounds appearing, two which were not in all the other genotypes. So this explains how the system is working. Let's look at methyl salicylate. This is a very important compound. If you have done any lectures or you're working on plant pathology, you will remember that methyl salicylate is one of the volatiles which is eliciting the so-called systemic acquired resistance in plants. So it's a volatile that plants do not produce all the time. When you do it, you are giving a signal to yourself and plants around that you're being attacked by pathogens. So you don't want to have that all the time. This is like being sick every day, like if having fever, right? You don't want to have fever all the time. And as you can see, in compacta, it is active, and it's also active, but in not in all plants, in the clock silence lines. So again, you see a compound, in this case, is a single gene required for you to activate the pathway. And that means that the coordination of volatile production from constitutive and non-constitutive is done at the genetic level and the pathway level. Look at this last one, which is called acetophenone. Acetophenone is a very interesting compound. It's produced in the flower, in most, and also in leaf volatiles, and it's a repellent. So it is, many insects don't like it. So you always produce a small amount. You may produce more and repel insects, or less, so that they will come to you. But it plays a key role in defense. And as you can see, it's active everywhere, but in compacta, it becomes non-constitutive. So compacta is probably going to have a problem in defending itself against insects. That compound should be there all the time, and is not. So constitutive components are sometimes very important. This is a near last one slide I'm going to show you, which is for another family of compounds. These are terpenoids. And here we find, again, a similar situation. Here we have ocymen. Ocymen is a typical major compound of the Oecium basilicum. Basilicum is a very typical herb. You use it in India. It is used also in the Thai food. It is one of the basic herbs used in the Italian cuisine. If you eat pasta, they will eat, there is a vegetarian sauce called pesto, and pesto is a basic sauce in the Italian cuisine. And this vegetarian sauce is made out with bay leaves. And bay leaves produce a lot of ocimene. And as you can see, the wild type flower is producing ocimene and also the RNAi, but you need your organ identity to be there all the time, otherwise you don't find it. And look at this pinene. Pinene is not produced by the wild type. It's never produced, but it is produced in some, as I showed you before. So this makes more sense now. You are getting a de-repression of this pathway somehow. And that is why you have some of these compounds appearing, right? And then you have other ones like this one, which is boring. It's there all the time, but it's important. Farnesene is non-constitutive, but it appears in all leaves, in all tissues, I say. So, what are the take-home lessons of my lecture? Number one is that organ identity and clock define the number of volatiles produced by flowers. So a flower is not producing anything, it's producing a certain number of components, and that is what is giving the flower the personality. That's why all flowers smell slightly different. And you want to have your smell, not anything you can produce. You don't want anarchy, okay? The number of volatiles produced is small. You can produce a lot of them, but you have to keep a certain set. That's the one that you want to use. And the lesson for students is, first of all, do your experiments. And then do the interpretation of the data. You will find that many times your data say you are wrong. Now go back and repeat the experiment and see that you didn't make a mistake, you didn't blunder. If you blunder with your experiments, then it doesn't make sense. But do the experiment again and see it is correct. And if the experiment is done right, and you got data showing that you're wrong, then you are enlightened. That's the way science advances. Most times science advances because we find we are wrong. So finally, I'm going to show the people which is giving the money, which is the government of Spain. We have work done by Fedetti and Horizon 2020, and we started a new collaboration. I became the chair of the so-called Bionet chair in Plant Biotechnology, where we have launched a new research program using bioreactors to produce plant cells for the industry. We are doing there plant cells to produce antibodies and other high quality proteins for the pharma industry, as well as secondary metabolites, okay? The people working with us in the lab, I have to highlight Professor Julia Weiss. I have with her a long-standing collaboration, including 3 children. So it looks like our relationship is sort of serious. And then we have Dr. Marta Terry, Fernando Perez. They did PhD in our lab. Fernando is now a bioinformatician working somewhere else. Claudio and Raquel are finishing their PhD right now. Onurcan Özbulat, that comes from Turkey. Victoria Ruiz, which is now a postdoc in Salzburg, and Professor Navarro, which we're doing with him, work on artificial vision, which I didn't show today. And with this, I finished my seminar, and I'm ready for any questions you have. Now, if you want me, I will keep the, I will maintain this open so that you can address any slide you may have questions for. Okay?
Thank you very much, Dr. Marcos, for the brainstorming presentation. Now session is open for questions. Yes, we have one from Anjali. She is writing, how cloak effect perfumes extracted from wood?
Okay. I'm going to tell you experiments we have that we published with Narcissus, and we're publishing a second set now. And I will go from the general to the particular. In principle, we found that the harvest time of the day can have an impact on the set of volatiles which are produced by a tissue. Okay, so it's not the same if you extract volatiles early in the morning or early in the evening. I'm going to give you one simple example. I guess you know that some cucumber varieties have to be harvested early in the morning, because when you get them early late in the evening, they become very, very spicy. They become more spicy than chilies. This doesn't happen with every variety, but it happens with some of them. So I'm not sure how strong is the constitutive volatilome of wood. If most of the volatiles are produced at the same level through the day, or you have differences between day and night. But it is a very, very, very interesting question to ask, to take small samples and test if you would get a different perfume, doing it in the day and in the evening. So I would do an experiment specific for that type of wood, taking samples. And this is important to remember. There is a concept in the clock, which is what is called the Zeitgeber. Zeitgeber in German means who is giving you the time of the day. So the subjective time is when the sun comes up. If I tell you 8 o'clock in the morning, it doesn't mean anything. You have to go to your the time of the sun in Hisar, if you're based in Hisar, and see at what time of the day is your sun rising. So if it is 6:33, 6:33 would be time 0. If it is 7:21, is 7:21, is time 0. So I would take sample at time 0, which is the time when the sun is rising, starting, and then the precise time when the sun is going down. A different question is if you take your wood and after you cut it, okay, you store it. We know that temperature of storage plays a key role, a huge role on that. We have a paper coming out, which is sent to Frontiers, and we are in a second round of discussion with referees, where we show that when you put a living tissue at room temperature at 5 or 20, or you give it a temperature fluctuation, okay, it makes a big difference on the volatiles which are produced. So I sent back to you the question, if your wood is a living tissue, you may find differences. If it is a dead tissue, then maybe not. Okay, so that would be the general answer. But I would do the experiment. I think it is a very, very beautiful, interesting biological question.
Okay, next question we have from Varnika. She's asking, can a single mutation produce multiple volatile compounds? And if it can happen, how? I mean, what's the mechanism behind it?
So the idea that we have is that, I will go back to the slide. The idea that we have is that if deficiency was a single mutant in a transcription factor, and that gene is involved in creating the petal. So we are talking about a gene which is here upstream, and everything downstream is after that changed. So you have genes which are upstream in the cascade, and they are mostly transcription factors or signaling. And then you may have hundreds of genes downstream which are active or are activated or repressed. So if you think about compacta, which is a gene which is controlling the identity of the organ, I'm getting a different petal. That petal is not okay. So everything with the petal is modified. The size, the color, well, the color is not affected, but the size is affected. Do you have other questions?
Yes. Next question is from Manpreet. She's asking, can we use acetophenone as pheromones for insect repellent in other plants varieties, and how to surpass the cloak effect?
Right. So so acetophenone. So that is an interesting question. Acetophenone has been shown that it repels many insects. It repels rodents, but it also repels pollinators. So it's what is called a double-edged sword. You know what I mean? You have to be careful when you use it, because if you use an excess of acetophenone, then you will not get pollination. Okay, you will get very resistant to everybody, including pollinators. So that's why we find very interesting the idea that, and the data that I didn't show, that acetophenone is produced, at least in our hands, at a basic level, like this. All the time you produce similar quantities. Then during the day, you produce more volatiles which are attractant. And during the night, those attracting volatiles are not produced. So what happens is that the flower is saying during the day, I'm ready to you, please come to me, I'm beautiful, I need to get pollinated. And then it says, I'm night, no, I'm tired, I don't want to have anything with you. It's night, I want to sleep, okay? Now, this happens because the flowers we are working with, they are from a region of the world which are pollinated by day by bees and bumblebees, okay? But if you look at plants, let's say from South America, which are growing in the tropics, that volatiles are produced during the night, then the acetophenone is there, and the nice smelly volatiles that attract the insects are produced during the night. So the answer, my answer would be, if you mess with the clock, you are in trouble. You are in trouble. Now, it could be that you could engineer plants with a leaf-specific promoter to increase the amount of acetophenone produced. The bad news is that we have genetic evidence that the acetophenone requires 2 genes for it to be produced. You can look, I'm gonna type here so that you can download the paper and read it. So there is a paper from Victoria Ruiz Hernandez in Frontiers in Plant Sciences, okay, that we published. Is open access. You can read it for free. And there we showed by genetics that acetophenone conforms to a 2-gene model based on epistatic analysis, okay, on classic Mendelian genetics, which sounds, by the way, I'm a geneticist, and doing classic Mendelian genetics is the best experiment you can do. You get so much information for just one cross that it is incredibly powerful, okay? So you can download that paper and read it, and you will see there the evidence that it's 2 genes. So those genes are not cloned yet, as far as I know, but I think it would be a good biotechnological experiment to try to produce higher levels of acetophenone on leaves and see if we could get out of that a stronger resistance to pests, because as you all know, they're a real problem in agriculture. Next question.
Okay, next question is from Alka. She is asking, how can we quantify these volatile compounds if we aim on any compound of interest out of these volatiles?
Okay, so quantification is a big problem. I will tell you why. There is an ongoing discussion in the scientific community. We have a paper, I will write it again here. So we have this paper, Alkyne Plant Methods. You can see Ruiz Hernandez et al., where we describe with detail different methods to quantify volatiles. Now, why I say there is a problem? When you extract when you do your volatile analysis, in most cases we use a fiber where volatiles get attached. And once you have those volatiles attached to the fiber, then you go with that fiber to the gas chromatograph, and then you release them, right? And once you release them, you get your area of the peak, and you quantify that. On this paper, we show that you don't always need an internal standard or a standard that you buy, and that is important because it makes things a lot cheaper, right? And we show by statistical analysis that, and you can read it carefully, and I know Alka is a very smart woman, so she will understand it perfectly well, and she can get by mail. The idea is that there are several methods that you can use to do the calculations, but where is the problem? The problem is that how well a certain volatile attaches to your fiber for extraction is not a solved issue. So some volatiles bind very well, and some volatiles bind very bad. So most of the fibers are optimized for non-organic volatiles. If you have, for volatiles which are non-polar. So the terpenoids bind very well, methyl benzoate binds very well, but if you look at benzyl acetate, because it has an acetate and it's polar, it binds much worse than methyl benzoate. So you're underestimating the amount of benzyl acetate. And the reason for that is that the fiber that that the community uses, there are 2 or 3, but all of them have the same problem. They are optimized for looking at non-polar volatiles. And what I'm telling you is a complex story, because if you think about how the community works, the whole community is looking at non-polar volatiles, and everybody's happy. And nobody's looking at polar volatiles because we don't have a good way to measure them. So it's an open question, right? Now, once you have your peak in your gas chromatogram, then quantifying, you can look at the different methods. There is one which is quite good, which is very cheap, which is what is called N-nearest alkene. That means you use several alkenes which are not in the plant, and you put them as an internal standard, and you use that area of the internal standard to calculate in nanograms the nanograms from your compound. I don't know if that makes sense, but otherwise I could explain it with more detail. Okay, next.
It looks like we have more questions. We quickly take 1 or 2 questions. Tilahun asking, can we conclude that housekeeping genes are not affected by environment conditions at all?
That is the idea. The idea is that the scientific community has looked for invariant genes, and this is how to say that, this is mountain evidence. So let's think for a second about Arabidopsis. Okay, Tilahun, you have people that made Arabidopsis experiments with microarrays, and they did day versus night, and salinity versus non-salinity, and heat versus cold, and stress versus non-stress, and short days, long days, all the possible different combinations that you can imagine. And after 150 or 200 papers that people have done 800, 900 microarrays, then they come and say, oh, these genes, they never change. And these are the housekeeping genes. And they don't change whatever you do. Age, environment, genotype, they're all the same, okay? Now, this is something quite interesting. Geneticists tend to like genes, like the question this girl was asking, or this person was asking before. How come that a single gene can activate or repress so many things? Well, because it's transcription factor, right? But the story is that some scientists have dedicated their scientific career precisely to the definition of the housekeeping genes, because they are really important. They are the basis, okay? And yes, you do have genes which are in every cell at nearly identical levels. Next.
Okay, I'm taking this last question from Harikesh. He's asking, can we produce same volatile or fragrance and compounds by using artificial light instead of natural?
Yes. The answer is yes, of course you can.
Yes.
Yes, of course you can. So important. Artificial light is a very, let's put it this way. Artificial light is too vague. Okay, today artificial light means something very complicated. Artificial light means using LEDs, which may be, for instance, giving you the combination for high growth. For instance, these ones have been used to produce marijuana. Okay, and they are red and yellow somehow, and then plants grow like crazy, really fast. Now, when you want to have the conditions which are like in the like outside, then you need to have blue light, you need to have red light, you need to have infrared. So you need all these wavelengths. You can produce that with relative ease, getting an LED set of lights, which are giving you what is called sunlight. In most cases, you're going to miss the UV light. So sunlight LEDs which are sold don't include UV LEDs. Why? Because UV LEDs are very expensive. Okay, so to put things in perspective, if you buy an LED for plant production, it can cost you 20 euros. If you buy one which has UV, it might cost you 200. Okay, but yes, you can. But again, you have to do the experiment and see if you get it. But I say that the core volatile will be there. Next.
Thank you, Dr. Marcos. I think we are good for now. And it was really a productive session, interaction with the audience. Now I request Professor Ashok Chaudhary to offer a vote of thanks to the speaker and the audience. Welcome, sir.
Well, thank you, Dr. Rakesh. Good afternoon, Dr. Marcos.
Hi, good morning.
Yeah, good morning. Okay. Since we were past 12 now, so that's why I said.
Yeah.
Anyway, yeah. Well, it has been really nice to have you again and, have words and, a very scintillating lecture from you, especially on the scent and the perfume released by plants and especially rice, because and especially I'm happy to know that you are involved in clock genes. And again, because I have been working on clock genes myself, CCA gene in rice.
Right.
And we have shown that it can also do miracles by improving the seed size and ultimately leading to increased production. So we are working on that, and we published one paper in PLOS One last year. So anyway, let me just take this opportunity to propose vote of thanks. Firstly, I would like to thank our Vice Chancellor, Professor Tankeshwar Kumar, who is present here and who has graced the occasion. He is a very dynamic person who is always, steering us and helping us to scale new heights. He is very supportive and encouraging us. And sir, on behalf of the department and all my faculty and staff and students, I'm highly thankful that you have graced the occasion and you are present with us today. Your encouragement and a path for the progress and future success. So thank you, sir, very much. And I thank Professor Marcos for a very scintillating talk today. And he has highlighted the importance of ultimately affecting, because perfume and scent production is not only important for the plant, but also it is helping ultimately affecting the production, because environmental cues will be the important factors which will be affecting, especially in the gene expression and transcription factors. And there are a whole range of temperatures. Temperature, another very important factor, as he has explained.
Yes.
That it will affect whether the gene will be expressed or whether the gene will not be expressed. So as few temperature degrees variations can really affect, and it is yeah, I highly appreciate the many breaking stories which you have let us know and shared your experience on molecular biology of plants. Definitely I'm happy, and I'm very sure that all our students will be very new ideas they will be developing, and that will be sensitize them for undertaking future research, especially in this important area of biotechnology. I also thank my colleague, Professor Vinod Chokar, for organizing this webinar series for a week, and all my faculty members, my colleagues, and they have worked very hard. Technology for giving us opportunity. They have supported us not only in this program for the MSc program, but also in bioinformatics, Nano mission, DST. We have done quite good job, and especially this program has been supported by DBT webinar series, and I'm sure all our students will be greatly benefited. So I thank all my participants, all the students, all my faculty members who are today witnessing Professor Marcos' lecture, and I'm sure they're going long way to go further. With this, I thank everyone and wish good luck to Marcos. And we will wish him, good health, and probably see him soon next time.
Thank you very much, first of all, for the invitation. I think that I think that the human soul is like made out of different. It's like an onion, that it has different parts, and there is a part of my soul that belongs to you. Yeah. That is a fact. I think it was an incredible experience for me to be there, and it was a humbling experience to be in contact with so many nice persons, so much interested, and the way you approach life was game-changing for me. I think, I think that I hope that my seminar was useful for undergraduate students all the way up, which is not easy. And again, anytime you need anything from me, I will be ready to help you. And I hope things will change and we'll go back to not the get a video for every person that took place that was taking the part in the seminar with 50 push-ups. I think Vinod Chokar said everybody was going to do 50 push-ups today for me.
Great. Yeah.
Thank you, sir, for the nice words.
Thank you, Professor Marcos. Professor Vinod Chokar.
This brings us to the end of current session. Thank you, everyone. We appreciate you being here. Thank you again for joining us today, and we'll inform you about the upcoming session. And we'll see you soon in next webinar series, next session. Thank you.
Thank
Have a
you
good
so
day.
much.
Thank you. Bye-bye.
Bye-bye.